Hey guys, welcome back. Now we've just produced pyruvic acid in process of glycolysis. What do we do with it? Well, the process in pyruvic acid can have two different features to it. The first being anaerobic. Now, anaerobic process in pyruvic acid is going to be in the absence of oxygen, and it's going to ensure that we minimize the effects of the acid buildup inside of our cells. And the other process would be aerobic processing of pyruvic acid. And in that case, what we would say is that in the presence of oxygen, we can take pyruvic acid and simply transfer it into the mitochondria where it can be further processed by the Krebs cycle and the electron transport chain. Let's first take a look at anaerobic respiration. If oxygen is not present, pyruvic acids will be converted to a more water-soluble molecule called lactic acid. And this lactic acid at rest will be transported to the liver, where it can be converted back to glucose. Prolonged anaerobic cellular respiration results in the buildup of acid molecules at the cell level, resulting in enzyme denaturization. Think of it. When would we see that happening? We would see that if we're exerting, for instance, our muscles in an environment that is anaerobic. We get muscle cramps, and those muscle cramps are due to the lack of energy production because the enzymes have been denatured from the buildup of acid at the cell level. This conversion involves transfer of hydrogen ions from an NADH molecule to a pyruvic acid molecule. Now, the end result then is going to be the production of lactic acid. Now, this is important, though, if you think about it, because it will free up the NAD molecules so that they can continue to be used during glycolysis. Please appreciate that NAD molecules are limited, and without them, the hydrogen ions would float freely in the cytoplasm. Now, what would happen if we had hydrogen ions floating freely in the cytoplasm? Well, think about it. What was the thing that caused the pH to decrease? It was an increase in the level of hydrogen ions, if you recall. So any free hydrogen ions floating freely inside of that cell, it would result in localized acidic conditions, causing enzyme denaturization and shutting down glycolysis. Now, the reality is glycolysis is an anaerobic cellular respiration process, and it is actually about 20 times quicker than aerobic cellular respiration. Aerobic cellular respiration would be when we took a glucose molecule through glycolysis, the Krebs cycle, and the electron transport chain. Aerobically, we can combust a glucose molecule and generate 32 ATPs. Anaerobically, we generate 2 ATPs per glucose, but we do it 20 times quicker. This means I can generate 40 ATPs anaerobically in the same amount of time I generate 32 ATPs aerobically. Thus, those muscle cells we looked at just a moment ago that need energy quickly, but for only a short period of time, example being somebody like a weightlifter, would use anaerobic pathway primarily to generate energy. Now, remember, if we do it anaerobically, we have a buildup of lactic acid. So typically, these muscle cells are going to be much larger. This is why we would see those bulky muscles of weightlifters and short distance sprinters. Now, on the flip side, we have the presence of oxygen. If we have the presence of oxygen, we're talking about something known as aerobic cellular respiration. Now, a set of reactions that are involved in aerobic cellular respiration are frequently called a transitional phase. Now, the transitional phase is going to include three reactions. If oxygen is present, the two pyruvic acid molecules will be actively transported into the mitochondria. What does actively mean? It means it's going against the concentration gradient. It's going from a region of lower concentration to a region of higher concentration, and or it is requiring a carrier protein in the presence of energy in the form of ATP. In this case, it requires a transport protein and ATP to bring those pyruvic acid molecules into the mitochondria. Now, each pyruvic acid molecule will be catalyzed by a multi-enzyme. Now, what is a multi-enzyme? That's a great example where we have more than one enzyme working in a cooperative way. Remember quaternary structure? So we have a multi-enzyme that results in first decarboxylation. The first thing that will happen is we'll take each of those pyruvic acid molecules and we'll cleave off one of the carbons. Now, anytime a carbon's cleaved off, it's going to produce carbon dioxide. The end product is going to be a two-carbon molecule called acetic acid. Now, once acetic acid is created, two-carbon molecule, it will be oxidized. Oxidized means that a hydrogen ion will be removed from it. And it will be oxidized, and that hydrogen with its high energy electrons will be used to reduce an NAD molecule. That means that it will take an NAD molecule and reduce it into NADH. And finally, that acetic acid molecule will be enzymatically bonded to a coenzyme A molecule. Now, what is coenzyme A? Coenzyme A is a sulfur-containing compound derived from vitamin B5. The end product of the transitional phase then is going to be an acetylcholine. Now, it's important, though, to remember that we have two of these pyruvic acids to carry through this process. Thus, we generate net, two NADHs and carbon dioxide molecules per glucose molecule that's brought in. So let's look at an overall summary here. The overall summary that we would have from glycolysis and transitional, we would see in glycolysis that we produce two ATPs, two NADHs, no FADH2s, no CO2s. We break one covalent bond, and the end product's going to be two pyruvic acids. Now, transitional, as we just seen, is we produce two NADH molecules and two carbon dioxide molecules, while we break two covalent bonds, and the end products will be two acetylcoenzyme A molecules. Thus far, then, we've generated two ATPs through substrate level phosphorylation. We've generated four NADHs that we will carry to the electron transport chain, and we've created two of the six carbon dioxide molecules that we're going to generate while breaking three of the five covalent bonds that were found in a glucose molecule. Next time around, we'll look at the Krebs cycle. Y'all have a great day.